I'm going to tell you something which is a story. And I'll start by sharing my slides. I'm going to speak on what's called diabesity in Indians, which is a combination of risk for diabetes and adiposity, a life course evolution. That means how it starts very early in life, how it evolves during your life, till it manifests in the phenotypes which we are interested, like say diabetes or obesity. And I'm going to base my talk almost entirely on the studies we have done in Pune. So it's a story of Pune studies. I work at the KEM hospital in Pune. I have a number of other attachments and a whole lot of collaborators from India as well as from abroad. And I have to say that mine is the only diabetes unit in the world where the logo is mother and baby, as you will see in the center of this introductory slide. There is a new logo which we have obtained, and that's the modern Ganesh. You can see this modern Ganesh. The seniors in the crowd, seniors in the audience will remember that when we were children, Ganesh used to travel on the back of the mouse. Given the disparity between the mouse and that's the vehicle and Ganesh, which was the passenger, there was a very slow travel. But the modern Ganesh now clicks the mice of his computer and he moves very fast. And that to me is the transition that has happened in India over the last 50 years. And therefore, I'm going to try and tell you a story that what happens when you are growing in mother's womb, which we will define as intrauterine programming, and rapid transition in later life, combined to give you the non-communicable disease. I just now heard some of the last few questions which you were asking, Dr. Chaturvedi, and there was a question about conflict and plagiarism. So my disclaimer is that I am only a clinician and a clinical researcher. I have not been trained in research methodology like you are being trained. I was so impressed by what was being discussed. I wish I was exposed to such things as a student. Unfortunately, I wasn't. So I'm only a clinician and clinical researcher. So you will hear from me a clinical research story. I have a bias because of my research in this area for more than 30 years that only life course studies should now be funded. Now you might disagree with this, but this is my bias. Cross-sectional studies can give you quite misleading answers. And I'm confounded because I always fall in love with my own hypothesis. So which is what a scientist is not supposed to do, but that's what I do routinely. Otherwise, it's impossible to carry on and on in a cohort like I will show you. Okay, it's not moving the slide. So it's also a story of how a diabetes specialist, what we call diabetologist, became a DOHAD scientist. And DOHAD is Developmental Origins of Health and Disease. It's a relatively new branch in medical science, new branch in basic science, and you will see how it's important to actually explain a whole lot of etiology of non-communicable disease and probably also of the infectious disease. This is the person who trained me in Oxford, Dr. Derek Hockaday, and his wife, Dr. Judith Hockaday. Both have been my mentors. Unfortunately, Mrs. Hockaday has passed away, but they actually saw my interest in investigating diabetes in Indians in a deeper manner. So I actually let my position in Oxford go and came back to India. And he then told me this story, which I realized later on is written by Pablo Coelho in The Alchemist. And basically, he said, when you want to do something, all the universe conspires in helping you to achieve it. And as you will see, what I have done over 30 years, setting up long cohorts, was possible only if other people had helped me. And that's what happened. So I'll start with this story of nutrition in diabetes because this is what was the story when I was a student. So that time, in the report of WHO on diabetes, in 1965, they said the evidence that malnutrition protects adult populations from diabetes seems unassailable. So everyone thought that diabetes was a disease of overnutrition, which is what we still think because we look upon an adult who is diabetic and more often than not, they are overnourished or obese. But a whole lot of research from India and other tropical countries came to the front in 1970s and 80s so that the 1980 WHO committee said malnutrition is probably a major determinant of diabetes, a complete turnover. And that's when actually I was training in Oxford in 1981 to 84. And the committee also defined an entity called malnutrition related diabetes, which was very ably described from Orissa in India by Professor B.B. Tripathi and his colleagues. So it was a boiling pot, and there was a scope to research in this. I think I have contributed something to the understanding of this conundrum, whether it's the undernutrition or overnutrition, and we will see how it goes. This, of course, led to a meeting in Orissa, which Dr. Tripathi organized, and we coined the term malnutrition modulated diabetes. But despite our efforts, WHO in 1999 classification withdrew this entity because they thought the evidence was not good enough. I am now trying to convince everyone that there is an evidence for undernutrition contributing to the risk of diabetes. So when I came back to India, suddenly the things became very difficult. I applied for a pool officer's post. I got the pool officer's post. So I started working in a government hospital in Pune, but that was not the place to set up long-term studies because in government hospitals, people come for acute problems and once treated, they go back to their original place and hardly ever come for follow-up. So I was trying to argue with CSIR, which is the Council of Scientific and Industrial Research, that I should be placed in a hospital where long-term studies would be possible. And I had this KEM hospital in mind because of its long history. And they kept saying no, it was 1.5 years, many applications, and the usual answer would be no, that's not possible. In 1985, Rajiv Gandhi went to US. He was our prime minister that time. And fortunately, one evening at 9 o'clock, I was listening to the Doordarshan news. And Rajiv Gandhi's speech from US to the Indian scientists was there. And he said, scientists who return to India will be placed in the institutes of their choice. So next day, I wrote this letter to Rajiv Gandhi. So a junior pool officer wrote to the Prime Minister. And within 3 weeks, I received this letter from his personal secretary, which directed the chief scientific advisor to place me in the KEM hospital. This I consider as a great victory of a junior person trying to establish, not sort of being helped by the system, but then the Prime Minister came and helped me. And this story I'm very fond of telling because if you as a junior are facing difficulties, don't give up. Try at the best places and at the proper time. And then in 1987, I got lucky because I got my first international research grant from the Wellcome Trust in collaboration with my boss in Oxford. And that was to, like, say, characterize metabolic and endocrine characteristics of Indian diabetic patients. That time it was for British pound 62,000. So today it would be something like 60 lakh rupees. But that was a big thing. It was for 3 years and it allowed me to settle. And I thought in 3 years I was going to solve the problem of diabetes. Now, 35 years later, I think I have now understood what is the problem. And that is again an important understanding that you can't solve important problems overnight. You need to really persist and it develops over a period of time. So I settled down in King Hospital, and this is now a 110-year-old hospital. And I was enormously helped by the founder of this hospital, Dr. Mrs. Banu Koyaji, who I consider as a great mentor. She let me work there. She said, I have a hospital, I have patients, but no money. So she said, you raise the money to set up your diabetes unit and your research. I had got this seed grant from the Wellcome Trust. So then I thought I would actually approach different people for help. And ultimately I got lucky. So I got grant from Mr. H.K. Firodia, who was then the CEO of the Kinetic Honda. Then Sri Rahul Bajaj from the Bajaj Auto. And here you will see an interesting thing. This is Pandit Bhimsen Joshi. So I was treating his family members. So he did a charity show for us to raise money for my unit. And all this put together allowed me to construct the top floor of the building here, which is called the diabetes unit. So again, if you want to do something, you have to actually go out of the way and make it possible. So that is about part of this story, which is not usually what you will expect to hear in such a talk. But I thought it's important for juniors to know this because with current situation now, grants are going to become even more difficult. So you will need to dig into many alternative sources. This was the first paper we published in 1991 from the Wellcome Diabetes Study. And what it said was that central rather than generalized obesity is actually related to high glucose in the Asian Indian subjects. And you can see here the two maps or two graphs about BMI, which is a measure of obesity, body mass index, which is weight divided by height square. WHR is the waist hip ratio. And I was the first one to measure waist hip ratios in Indians because that time it was a very new thing. The concept of central obesity had just been described from Sweden. So when we did this, it was quite amazing that people with lowest BMI but highest waist hip ratio had the highest blood glucose, whether it was men or whether it was women. So this was our first understanding that it is thin but centrally obese people who are likely to have higher glucose and therefore higher risk of diabetes. As anthropologists and related branches, you are familiar now with these measurements. You probably look at it in different way, and this will be a good food for thought for you to investigate what might be the anthropological sort of background to this phenotype which has developed in India. So low BMI, high waist hip ratio, a strong predictor of diabetes. And then I developed this concept because what I did was I compared patients in my clinic, that's in Pune, and patients in Oxford Clinic where I had done my training. And the difference was really obvious. It was obvious to the eye. So you really didn't know any or didn't require any rocket science to prove what was the difference. And this is a very simple figure I drew after comparing 200 patients from each site. Of course, I was measuring simple things. The first point was Indian diabetic patients were diagnosed with diabetes at least 10 years younger than their British counterparts. So diabetes appears in Indians at a younger age. And we know that diagnosis of diabetes happens years after it has started. So you can imagine that actually the time for that to happen was much before that time. Secondly, the body mass index, that's obesity index of Indians, was 5 units smaller than the English patients. So average something like 24 in Indians, 29 in the English. Mid-arm circumference, which I'm sure you measure in your anthropometric measurements, it's a surrogate for muscle mass. So Indians had about 20% smaller muscle mass surrogate compared to the English patients. So our patients were young, they were thin, they were lean. Muscle mass was less. But look at their waist-hip ratio. Despite all this, our patients had 5% higher waist-hip ratio. Waist-hip ratio variability is subtle. And if a 5% bigger waist-hip ratio with a 5-unit lower BMI was a big thing actually. And I had also used the skin fold calipers. So we measured subscapular and tricep skin folds, and their ratio was higher, indicating subscapular is a central fat, triceps is a peripheral fat. So central subcutaneous adiposity was higher in Indians. Later on, when I got money to measure body fat as body fat, because it's not very easy to measure, we showed that Indians had 30% higher body fat percent for each BMI unit. So that was quite a revealing sort of description. I tried to publish this in various journals. There were a lot of national and international invitations for me to speak, so people knew about this idea. But editors were very resistant to accept what I called a thin fat Indian. They thought it was very counterintuitive and address Pune, India might not be Oxford, UK. So I faced a lot of struggle. Finally, I convinced everyone when I sat on a WHO committee, which was invited to define body mass index cut points for different populations. And we wrote a 6-page, very erudite report where the world's best people were there to write that report. And I said, I have a picture of myself and my friend. So you might find it difficult, but 30 years ago, I looked like this, like a handsome young man. And my friend was here. We were doing a study together, and we thought that whatever we expect the participants to do, we should do it ourselves, which means we should measure ourselves. And to our great surprise, what we found was that Mr. Yudkin, that's my friend, and Mr. Yajnik, that's myself, we both had identical BMI, 22.3. That was purely by chance. 2 principal investigators in a study having identical BMI. Then we went on a machine which is called DEXA, dual X-ray absorptiometry. That machine gives you body fat percent. John Yudkin's body fat percent was 9%. And my body fat percent in 1999 was 21%. So for same BMI, An Englishman had a body fat percent of 10%, and I had a body fat percent of 21%, more than twice as much. So that picture convinced people that what I was talking about, thin fat Indian, might be right. And that's why this picture came in Lancet, which is a very prestigious journal. It's called YY paradox. That's Yajnik and Yudkin paradox. And this picture is one of the most frequently shown in all diabetes and obesity meetings since it was published in 2004. So you will see internationally I am called a thin fat Indian. So that's the story behind this phenotypic definition. And you should realize that one picture, they say, is worth more than 1,000 words. So if you have an idea which you want to sell, and you can't write it properly, go after a very revealing picture, which will reveal the content of your idea. So now we come back to India again. And India, as you know, is called world's diabetes capital. China has more patients than India, but India is the summer capital of diabetes. And that's how in this graph, which is called a cartogram. I'm not sure whether you are familiar with this form of representation. Cartogram is a map where the size of the country is proportional to the burden of that disease. Not to the geographical area, but to the burden of the disease which you are describing. So if you look for diabetes, India is one of the fattest countries. Is India even fatter for something? It's here. It's for undernutrition in early life. So low birth weight. About 25% of the babies born in India, even today, may be low birth weight. So every year we give birth to more than 25 million babies. 1/4 of them are below 2.5 kg, which is the definition for low birth weight. And then world's largest number of under 5 undernourished children are also in India. So you have this paradoxical comparison here. India is the fattest country for diabetes, but also for undernutrition in early life. So that's a paradox. We are capital of two conditions which are counterintuitive, undernutrition and an overnutrition condition. And then everyone told me that this must be genetic. Now, yes, that is possible. Today it's possible to do high throughput genetics, work out all the SNPs related to this, that, etc. So yes, that's possible. But up till now, we have not succeeded in showing that the inheritance of adiposity and diabetes is substantially different in Indians compared to other populations. So what really might matter and what might be important is that yes, genetics must be playing a role, but what is not genetic? Everything is genetic ultimately because we start with a bag of chromosomes. 46 chromosomes, half coming from father, half coming from mother, and that's how we begin our life. And then a whole lot of things, and you'd evolve into an individual. And that today is called epigenetics. So epigenetics is involved in the human development, human differentiation, and now we know in predisposition to a number of non-communicable diseases. And the first dramatic example of this was published sometime in 1930s by two veterinary scientists, Walter and Hammond. And what they did was, there is a Yorkshire horse, which is a very tall and hefty horse. And this is a Shetland pony. Many of you will know Yorkshire is in England. Shetland is northernmost islands of Great Britain. Very harsh conditions. So animals are also exposed to a lot of environmental stress, malnutrition, etc. What they did was they mated a Yorkshire horse with a Shetland pony. And what do you think would be the phenotype of the offspring? Would it be tall and hefty as father, or would it small and stunted like a pony? So answer is here, this is father and daughter. So the daughter was more like mother. And this was the first demonstration that though equal genetic input came from two parents, it was probably the environment in the uterus which would determine the child's phenotype. So this is a brilliant example from veterinary sciences. And then came quite a dramatic event in my life. That was February 1991. That is when Professor David Barker, shown here, visited me for entirely wrong reasons. I mean, he happened to be in Pune for some other reason, but he had made a very great discovery that in England, in a cohort born in 1930s, he demonstrated that lower the birth weight, higher was the risk of diabetes in 60s and 70s when he measured them. So you can see here, as the birth weight increases, the risk of diabetes decreases. So he had formed this theory that low birth weight is a risk factor for diabetes. Till that time, I knew that India was actually suffering from low birth weight problem. I knew diabetes was there. We had never connected it. David Barker connected that for us. And when he told me his story on a February morning in 1991, I suggested this term for his hypothesis, Thrifty Phenotype. And he wrote that paper 1 year later. He said type 2 diabetes is the outcome of the fetus and early infant having to be nutritionally thrifty. That means that food supply was small and the fetus had to make the best of that food supply so that A, it doesn't die in utero, so it survives, and B, it survives in such a way that he comes out alive and start growing. So that is a thrifty phenotype. And what happens at that time? In this short-term decision to survive and come out, fetus has to make a series of alterations to its structure and function of the developing organs. And the price he pays is much later in life when there is excess environmental, nutritional, and physical activity or mental stress, that he is born with low capacity organs, and when the load becomes too much, the organs do not perform well. So that is the thrifty phenotype hypothesis. On a February morning in 1991, it completely changed my idea. So rather than thinking of genetic explanation for Indian phenotype, I started thinking that this might be the explanation. And I told David Barker Abhimanyu's story. So you can see Abhimanyu's picture here. All of you know Abhimanyu learned how to enter Chakravyuha when he was growing in his mother's womb. So Krishna was telling the story to Subhadra. Abhimanyu heard it. The story was stopped halfway. So Abhimanyu never got to know how to come out of the Chakravyuha. He knew how to go in. So during the great war of Mahabharata, he was the only one who was able to go in, but he didn't know how to come out, and that's how he had to die, basically. So I told David Barker, we have a great example of brain programming in Indian mythology, and I suspect David Barker actually started believing his ideas more strongly after knowing about Abhimanyu. So now we can see that we can connect this paradoxical distribution of two conditions related to overnutrition and undernutrition through mother and the baby. And that is the focus of my research since 1991. History always doesn't leave you. So you need to learn a lot from history. And this is a very dramatic graph of height in different populations of world, 3 European countries and 3 Asian and Southeast Asian countries. Between 1820 and 1980. In these 150 years, Europeans gained 15 centimeters in height. So 1 centimeter for each decade. How much did Indians gain? This yellow line is Indians. Hardly anything, 0 to 1 centimeter. And many populations in Southeast Asia had actually lost height and subsequently recovered part of it. So this history suggests that for last 200 to 300 years, we have faced tremendous environmental stress, nutritional stress, many famines, many floods, a whole lot of things, and that has suppressed our linear growth. After 1980, things have dramatically improved. But this is our history for last many generations. And this map of India is a seminal work published by Public Health Foundation of India as part of what is called their Global Burden of Disease India investigation. So Professor Lalit Dandona, I am one of the authors on this paper. And what we showed was that the rise in diabetes prevalence in 25 years from 1990 to 2015 was different in different states of India. Not all states are equally affected. So the red ones and orange ones are the states where maximum rise has happened. And they are predominantly something like Bimaru states. So these are the states which have suffered deprivation for last many decades, if not centuries. So Uttar Pradesh, Madhya Pradesh, Chhattisgarh, Odisha, then Nagaland, and Arunachal Pradesh, Jammu Kashmir, Himachal Pradesh. These are the states which showed maximum rise between 1990 and 2016. Again, indicating based on what I'm showing you here, is that a small amount of development which has happened in 25, or you might say a substantial amount of development, the population could not tolerate. Very likely because they all had thrifty phenotype. Because we don't think genetics can make a difference in one generation or two generations. So that's a very nice combination of two graphs that actually support the idea that undernutrition followed by overnutrition actually is very detrimental for your body phenotype and for your diabetes risk. So great lesson to learn. 3 years ago, I did a visiting fellowship in Denmark as a visiting professor to Danish Diabetes Academy. And Soren Kierkegaard is a Danish philosopher. And that time my great learning was that he said life can only be understood backwards. And this is true for all of us, but certainly true for evolution of a condition in a population. And therefore, I would urge you that if you want to study something today, an outcome, always look backwards. And looking backwards is possible in cohort studies because you have collected serial information. And this is my record from 1987 to 2014. We have set up a series of cohorts. And we have answered very many different questions from each. I'm not going to get into this because we will be then talking till evening. But I'm going to concentrate on one cohort called Pune Maternal Nutrition Study because that is the flagship of my research and flagship of my department. You can read this, the reference is here. So Pune Maternal Nutrition Study is the first pre-conceptional birth cohort in India. I don't know of any other birth cohorts in India which are pre-conceptional. And this was set up to find out determinants of fetal growth in India because Indian babies, as I said, are the smallest in the world with a birth weight only on average birth weight of 2.7 kg. And to study life course evolution of phenotypes because in a birth cohort you can do that by studying the mother, then baby when it is growing, and then following up the baby, mother and the father over long periods of time. So we set up Pune Maternal Nutrition Study in 1993. In 2019, we started 24-year follow-up of the study. And in February 2020, I had to stop because of COVID. So I will have to wait for COVID to go away to start again. So how did it happen? So we went to 6 villages near Pune, and these 6 villages were that time in what we called a drought-prone area. So they didn't have much water supply, they had a drought, they had famine, and populations were quite poor. The original design was we went to 6 villages and recorded every house to house survey. We gave each house a unique number, which was an 8-digit number, which incorporated all the information we needed. Which village it was, which wadi vasti, which house number, etc. 8-digit description. And we detected that more than 2,500 women were there who were married but not pregnant. And my interest was to set up a birth cohort. So we needed women who were likely to become pregnant. And that was a number more than 2,500. So we followed them every month for their menstrual dates. And every 3 months, we did their detailed anthropometry as a surrogate of their nutrition. During 3 years of study between 1993 to 96, 814 women became pregnant and delivered in our study. We studied them in detail 3 times in pregnancy for their anthropometry, nutritional intake, physical activity, metabolism, blood pressure, fetal growth by ultrasound, and father's size and father's metabolism. So a very detailed study 2 to 3 times in pregnancy to document maternal nutrition and physical activity and other aspects of sort of exposures which the baby gets in pregnancy. 770 babies were born in our study. We measured them in great detail for anthropometry. So we not only measured their weight, but we measured their length, their head circumference, chest circumference, abdominal circumference, mid-upper arm circumference, and skin folds. So a very detailed phenotype. Then we have measured these babies every 6 months till they became 18 years of age. And after that, we measure them once a year. And every 6 years, we study the children and their parents, again for their size, body composition by a DEXA machine, sometimes MRI, nutrients in the blood, cardiometabolic risk factors, cognition, a whole lot of things. There are many things which may not be mentioned here. And you can see our follow-up rates are phenomenal. More than 95% till 18 years of age. And at 24 years of age, I think this is by mistake, it's a cut paste mistake. We have been able to study up to now 250. So at the end of the study, we will know what is our percentage follow-up, but that will have to wait for COVID to go away. Now, during this time, we have also established a biobank so that each time we studied mother in pregnancy, we took a blood, urine, and we have put it at minus 80 degrees centigrade freezer. We collected blood samples at each of the follow-up visits. So we have a unique biobank which starts from woman's pregnancy and goes through her subsequent follow-up and her child's follow-up. Okay, I don't know, it's not moving forward, sorry. I'm just going to come back to that again. So this was the area. 6 villages, about let's say 30 to 70 kilometers away from Pune. And this is the distribution of the subjects in these villages. So that was the geographical background. Within last so many years, now 140 people have migrated within the district for either education, work, and marriage. And we follow every one of them. That's why we have follow-up of more than 95%. So if you want to set up a cohort, follow-up rates are very important, and you have to be prepared to pay extra efforts and time in that. These are some of the quite, like say, nostalgic pictures for me, how things happened at that time. The atmosphere was quite pristine. So first thing you see here is the village head, and we had to first obtain his permission to do the study. In addition to ethics permission from the hospital ethics committee, we had to obtain a village leader's permission. Without that, things would have been impossible. Then we went for consent or assent from the participants depending on their age. And we employed local, like say 10 standard pass girls as ANMs, or some which had done the ANM course who had a higher sort of appointment. So we employed a dozen of them, and there were 4 supervisors which were male. They were also trained in anthropometry. And for these village sort of ANMs, we gave a bicycle and a bag which contained all the tricks of the trade. The equipment for anthropometry, then books for recording it, and whole paraphernalia that was required. And we also developed a mobile van with a fitted ultrasound machine and a small laboratory, so that every day a team from Pune, including an obstetrician, an ultrasonologist, driver, and a social worker, visited one village. So if one village on Monday, Tuesday, 6 villages by Saturday, again start next Monday. And within the ultrasound, within the van, we could do ultrasound. That time the PNDPT Act didn't allowed us, or there were no PNDPT Act. Therefore, it was possible to do a mobile van ultrasonography. We also visited the women early in the morning when they were fasting to do their glucose tolerance test. Then our nutritionist visited them at 3 times in pregnancy to take down their nutritional intake. And you can see here how in the field, 2 of my nutritionists are taking down her daily food intake while she is working in the field. And when they delivered babies, we did detailed measurement of the baby 50% of the deliveries happened at home. So we did it at home, and others happened in primary healthcare center. And we had a fantastic epidemiologist in form of Dr. Hirve, who conducted and supervised all these studies. And he is now a leading epidemiology officer employed in WHO. And you can see I interacted with the community quite well. So you can see me here with a feta given to me, talking in one of the meetings of the patients, not the patients, but the participants. And we had an inter-observation variation study regularly, given that 4 people from village who were trained were measuring. We had actually coefficients of variations of 1 to 2%. In other studies where a doctor was measuring, it was lower than this, but we thought this was acceptable. But it's important to ensure quality control. And then data management was quite substantial. So we had actually an advisor, Dr. Poonam Gupta from the National Institute of Informatics, so NIC. And she actually helped us for the data management. So it's a great achievement. In these days, computers had just started functioning in public health studies. So we did actually all this on computers. So it was not like today, it was the older generation hardware and software. And then this is the biobank which I mentioned you. This is the most expensive investment I have done. No grant gives you money for biobanking. So today I have 25 freezers with me, in which there are more than 500,000 tubes which are stored at minus 80 degrees centigrade. Electricity bill is huge, but this is a unique resource which will allow us to do a whole lot of research in future, even when I'm not there. So the average mother in our study was 42 kg with a body mass index of 18. And those of you who are familiar with this, 18.5 and below, WHO calls undernourished. So these women, 60% of them were undernourished by WHO criteria. Their babies were born at an average of 2.7 kg, and by Western criteria, 70% of them were SGA or small for gestational age. But look at this mother. She is 7 months pregnant. She is 47 kg. She is carrying these 2 pails of water from the village river. Each one is 15 liters. Her neighbor is carrying 4. So you can see that though they were thin, they were eating less than recommended food, they had a substantial physical activity in various activities of the home. And there was very little gestational diabetes, less than 1%. This is not true today. Their daughters have 20% gestational diabetes. We are not going to go into next generation much, but that is the rapid change that has happened. And these babies at 2.7 kg looked very thin. But I had my experience from before about being careful in calling someone thin without measuring body fat. So we did a detailed anthropometry, as I mentioned you, and in a limited number of babies, we did whole body MRI, which allows us to measure different things very carefully, especially abdominal fat. So what did we find in anthropometry was when we compared it with a 3.5 kg English baby, the smallest component of the Indian baby's measurement was abdominal circumference. So it was almost 2.5 standard deviations smaller. Then the weight and length, etc., were 1.5 standard deviation smaller. And surprisingly, the most preserved measurement was subscapular skin fold, the central fat. So you can see in this baby, there is a hierarchy of smallness. subscapular kinfold of our babies, even though they were 800 grams lighter, was almost similar to the English baby. And when I did the MRIs, we realized that actually our babies had more fat in the abdomen compared to the English babies, even though they were 800 grams lighter. Their cord blood also showed that levels of a hormone called leptin, which is secreted by fat cells, was higher in the cord blood of the Indian babies. So this is a very remarkable description of thin fat Indian babies. And again, lot of resistance for publication. But now this is a very well-known concept in DOAG and everyone quotes it everywhere. And a lot of maternal nutritional factors influence baby's growth, especially green leafy vegetables, milk and fruit, three sort of food items which are rich in micronutrients. Calories and proteins had much smaller effect, probably because they were all eating small amount and very similar in different people. Red cell folate in the mother again had a relationship to baby's size. And if you have heard of homocysteine, which is one of the amino acids in the blood, which is produced by folate and B12 deficiency, high homocysteine was associated with increased risk of SGA, that means small baby, that means fetal growth restriction. And to our great surprise, these high homocysteine levels in the mother came from B12 deficiency, which was present in 2 out of 3 mothers. Folate deficiency was almost non-existent. Homocysteine was high in 1/3 of the mothers. And MMA, which is a specific measurement for B12 deficiency, was high in 90% of the mothers. So we had established a finding that Indian babies were small, thin, but fat, and their growth was affected predominantly by micronutrients in the mother and one carbon metabolism, which is what homocysteine represents. If it is deranged due to B12, folate, B6, B2 sort of vitamin imbalance, then the baby had a high risk of being born small. And in addition, mother's glucose, cholesterol, triglycerides also affected different aspects of baby's growth. So there is a very detailed description. I'm not going to get into details, but what we are showing is both maternal macronutrients and micronutrients contribute to fetal growth as is expected. But the micronutrient story and B12 folate story is a new one. And what happened when these babies grew? So when these babies grew, our whole idea was to see whether maternal nutrition in pregnancy affects baby's future. And certainly it did. What we found was that at 6 years, baby's adiposity, that means fat in the whole body as well as abdomen, and baby's insulin resistance, which is a precursor to future diabetes, was high in the babies who were born to mothers who had low B12 and high folate concentrations in pregnancy. In other words, an imbalance of low B12 and high folate was somehow or other programming the baby such that it became more adipose and it became more insulin resistant, two factors which will predispose it for future risk of diabetes and adiposity. And recently we have done MRIs of these children at 22 years of age, and we have done a whole lot of neurocognitive testing as I told you. And what we find is intrauterine exposure to different micronutrients has affected development of certain areas of brain in these children. So this is a high resolution MRI image and a functional MRI connection, which shows that certain areas of brain are underdeveloped if your mother had deficiency of B12, and especially if it was associated with high levels of folate because the national government programs provide iron and folic acid, but not B12. So what was the situation when they were 18-year-old? At 18 years, half of them are still underweight. That is BMI less than 18.5. 5% are overweight or obese. That means BMI more than 25. 10% are stunted. And on the glucose side, there is a remarkable finding. 27% of boys and 8% of girls have high fasting glucose levels. 10% boys and girls have high 2-hour blood level glucose during oral glucose tolerance test. So that a total of almost 30% of these thin village young people have what we call in jargon pre-diabetes. So that's quite a shocking and amazing statistics. They are underweight and they have very high prevalence of glucose intolerance called prediabetes, that is twice as high in boys than in girls. And there are other associated findings that insulin secretion and its action is defective in them. Now the interest is, if you have prediabetes at 18 years, can we now find out when did it all begin? That is the Lifecourse analysis. And to our great surprise, or not to our great surprise, we found that those who have prediabetes at 18 years had higher glucose at 12 years and higher glucose at 6 years. That means it was obvious when we first measured them, but the current cut points are all concentrated toward adults and no one knows what is high glucose in childhood. So we just let it go, but actually it has all happened from very early age. And that is the strength of having a birth cohort and serial measurements of everything. And we also found that growth of those who were pre-diabetic compared to those who were non-pre-diabetic was different. So this dark black line represents the mean growth of a pre-diabetic child against the mean growth of NGT, which is shown as 0 line. So pre-diabetic girls and boys had a shorter height from birth, and girls continued to be short throughout their life, while boys actually are coming nearer to the mean line. Their weight, girls were underweight throughout. Boys were near normal weight but picked up weight so that some of them have put on weight during puberty. And BMI for girls consistently low. For boys, it was normal compared to LGT and has started picking up after puberty. So important learning is, again, if you are doing a study, you should study adequate numbers of men and women separately so that you are able to find out the differences in the sets. We have also done detailed studies. I'm not going to go into details of it, but we have measured how their pancreas functions, how their liver and muscles sort of respond to the insulin, etc. But in summary, what I'm going to tell you is that those who are pre-diabetic at 18 years and have their substantial differences in insulin secretion and action, all that is traceable back to 6 years. So that the abnormalities are there from very early in life. And then now you can imagine 25 years later, 200 girls in the cohort have married. They have become pregnant. So we have studied them through pregnancy and their children are being studied. So we started with F0 generation, F1 was born, and now we are studying F2 generation also. And very interestingly, if we look at glucose levels in the girl during pregnancy, we can track them back to her 18-year glucose, to her 12-year glucose, and to her 6-year glucose. So again, what we are seeing as so-called gestational diabetes has been sort of tracking since very early age. Though in current standards of practice, we don't call it abnormal because we don't know what is normal and what's abnormal. So this is the world's first study to track back the glucoses of girls. These young girls, average age at pregnancy 20 years, that their glucose in pregnancy is a manifestation of what was happening from their childhood. So we come back to B12 and why Indians have so much B12 deficiency. So without going into details, I'm going to tell you that it's due to vegetarianism as a substantial cause. Because B12 comes only from non-vegetarian food, and for that matter, milk is also non-vegetarian. So milk, eggs, fish, meat, they provide you B12. Plants do not provide you B12. Plants don't need B12 for their metabolism. They don't have it. So in India, vegetarianism, unlike in the Western world, is again multi-generational. And it started with Samrat Ashok, who became Buddha's disciple after the Kalinga War. The Buddha taught him about ahimsa, so not killing only the human beings, but not also killing the animals for food. These three religions, especially Jains, Hindus, believe in believe in vegetarianism. So there is a religious and cultural drive towards vegetarianism. And in a very interesting study, we found that higher education, higher income, and better hygiene predisposes you to B12 deficiency. And this is very interesting because B12 in nature is made only by microbes. So microbes make B12, animals eat it, and we eat animals to get it. But bacteria can also provide us, and you know the stories of microbiome, which we are not going to discuss today. So increase and obsessive hygiene might actually predispose you to B12 because you might then also get rid of the useful bacteria. On the other hand, folate is a plant product. So folic acid, foliage is folic acid. So if you have an adequate vegetarian diet, you should get enough of it. If you have a higher socioeconomic status, you should then get enough of it. The National Anemia Prophylaxis Program provides you iron and folic acid to adolescent girls, pregnant women, and women in reproductive age. And obstetricians use high-dose folic acid to prevent neural tube defects. So we put a lot of folate and folic acid into our system, either by your food or through supplements, when our main problem might also be B12 deficiency, and therefore we exaggerate the imbalance between B12 and folate. So this is a DOHAD model that you start very early in life before your conception, that causes changes in your genes called epigenetic changes that affects your fetal growth, development, differentiation, gives you a particular phenotype. Then on that phenotype, there is a subsequent environmental action. So it goes on evolving. Finally, we measure risk factors, morbidity, and mortality. So pregnancy, therefore, is an experience for the two. But I'm now going to tell you something quite dramatic, which some of you might have heard before. In certain pregnancies, it is an experience for the three. I don't know how many of you know this. This is a pregnancy with a female child. And a dramatic realization in last few decades is that a female child has all its ovaries, which is 1 million of them, which she is going to have for the rest of her life, are present by 20 weeks of gestation in utero. So if you are studying a pregnancy with a female child, or if you are intervening in a pregnancy with a female child, you are actually interacting with three generations. And this is a very dramatic thing. Anthropologically also, you can now start thinking about this. And I have really learned this. This was the picture in the last paper which David Barker published in 2013 before he passed away. So it's a paper in Nature. And epigenetics is at the root of this so-called fetal programming. There are various mechanisms. We are not at all going to go into this, but we are making detailed studies of these now in our cohorts. So the model which we have now of so-called heritability is that there are paternal and maternal genetic factors. And on the either side, there are epigenetic factors which continue from the mother's side during lactation. And then later on, there is a family environment in the postnatal life, which is influenced partly by biology, that's genetics, also by the family environment, so their diet, hygiene, education, socioeconomic status, then culture, religion, and outside this, you can draw national and international policy and the socioeconomic sort of movements which happen across countries, and that all influences the final phenotype. So with all this in mind, actually we decided to intervene in our F1 generation to improve their B12 status, and the questions were with what, when, and how. I'm not going to go into details of this figure, which is published in one of our publications, but it requires a whole lot of thinking and planning. So first we did a pilot study to give only a very small dose of B12, 2 micrograms, which is the recommended dietary allowance, to see whether we can reduce homocysteine substantially, and we were very successful in that. So we knew in deficient populations this small dose works, and we wanted to make our trial public health relevant, so we can't give large doses. And you can see here children which contributed to this pilot trial. They have their booklet in hand. They have signed their assent here. So they are very happy to display that they are part of this, they are active part of this. I told you about the Danish philosopher. Many of you would know about Steve Jobs, who was the CEO of Apple. And I found this quotation from him, which is quite stunning. He said, you cannot connect the dots looking forward. You can only connect them looking backwards. So you have to trust that the dots will somehow connect in your future. So that's quite a great statement for an intervention in a cohort where you have all the information going back, where you connect the dots, and you hope that this will work in future. So there's also another Lancet series about preconceptional health. There are 3 articles. I'm author on 2 of these. You should read this, but it tells you why preconceptional intervention is so important, because that is the time when epigenetic programming happens. So we have done this trial. I'm not going to go into details. It's called Pune Rural Intervention in Young Adolescents. It's called PRIA. And this happens in F1 generation in our cohort. We intervene both in boys and girls with either vitamin B12 alone, B12 with multi-micronutrients and milk powder to improve protein intake, or a placebo. And we have now stopped the intervention just before the COVID struck. And we are now analyzing the results. We are not going to be able to discuss that today. Small aspect about ethics of cohort research. And I think you are going to listen about ethics in other talks. But the first rule is do no harm. And then there are 4 principles of this. I'm not going to get into detail, but we spend a whole lot of efforts in getting the ethics right, getting informed consent. And on average, we have had 10 consent forms signed by parents from F0 to F1 generation at different times, and then children were signing assent, and whole thing was done very openly, and they really enjoyed it. So more details can be available with our personal discussion, but that's how we have done it. The informed consent forms were in local language. They were actually explained to them, and various other methods have actually been adopted. I think I'm going to stop now by telling you something even more interesting. I give these talks in many places. I get a lot of invitations now that people know about our studies. So last talk which I gave in Denmark was a European Society for Obstetrics and Gynecology. And this lady came up to me. She is the dean of this Polish medical college in Poznań. And she said, your name, Yajnik, is very strange for us. It struck me as a strange thing because she said in Polish, Yajnik means ovary. And she said, why I can see you were obsessed with investigating the programming in utero and the preconceptional, because your name has a background on this. So that's just for your entertainment in a way, but quite a profound thing for her to come and say to me. Such studies need a lot of funding, and without that, you are nowhere. So I have been lucky that many international organizations and national organizations have funded, but there are large chunks of time when no one is funding you. And I have no hesitation in telling you that I donated half of my clinical trial income to run the cohorts, because otherwise they would have stopped. So you need a very proper funding source, and you should be prepared to have personal sort of sacrifices to achieve this. So I'm going to stop here. I have told you the Pune story. I hope you liked it. It was nothing high science like what you might have expected, but it's very interesting. And if it stimulates some of you to set up cohorts and do the studies like we have done, you might be able to answer some questions which are never going to be answerable in many of the cross-sectional studies. So thank you very much.